Okay, so nothing is perfect, right? And we have what we call a base miss pairing. That actually means that instead of an AT or an AU pairing and a GC, we suddenly see some kind of mixture between the two pairs. That is, of course, against the usual restrictions, and it happens. It's a mistake. Um, it might be a result of an insertion of a pair into the parental molecule. Okay, so we see that too, but that is a mistake. The inserted base into the parental molecule suddenly allows another nucleotide to be paired with it, and thus changes into the complementary nucleotide. So it happens. It's rare, but it happens. So you are referred to a movie, and we were talking about in the introduction presentation about a sequence that is inserted into the original molecule. I gave you the example of alien, so to speak, DNA, but it doesn't have to be alien. It can just be a sequence that for some reason has been taken from another place in the cell, another molecule, and just inserted into this molecule. So those sequences is what we called additional stretches, and when those kind of stretches can be moved around, we call it transposable elements. Okay, so transposable elements, can move easily between sites of a certain gene. And we actually find that. We find sequences of the same gene moving from one place to the other. When that happens, most likely that the gene will become useless, or what we call abolished, meaning that it's going to lose its original role and function. Okay. Through evolution and even through different supervision mechanisms, quality mechanisms of the cell, those regions, those sequences, undergo what we call deletion. They're cleaved out. They're taken out. Okay, because they don't add up with what the cell knows is supposed to be in that gene. So you're referred to a movie. I hope you enjoy it. When we're talking about deletion, I didn't indicate how big the sequence that is being deleted. So sometimes it can be a really big chunk or a really long sequence, in other words, out of a gene that has moved and now is deleted. Okay. When we're talking about mutagens, they may affect point mutations, but they do not affect insertions or deletions, meaning that they cause mutations, but they're not the ones that decide if something is going to be inserted or deleted. Okay? Both insertions and deleterious mutations may occur during the replication of a molecule or a recombination of a molecule, right? So we have the DNA and we want to make the daughter strands. That's one process where something can be inserted or deleted, and the other one is when we have those two separate strands and we want to combine them, recombine them, right? That's the other option when it can happen. when we have mutagenes that cause an inserting or deleting sometimes, which is rare, we call them acridines. Okay, so that's a very specific option of mutagenes. Once again, we have mutagenes, those who are able rarely to cause an insertion and deletions are called acridines. Is a mutation final? No, it's not. Because here's the thing. We're talking about sequencing, and we're talking about the ability to insert something and also delete it, right? Cleave it out. So let's say that a mutation occurred, a point mutation occurred, and a sequence was deleted. Let's say there's a mistake in one base pairing. We just talked during a few videos, well, I talked about the ability to cleave it out. So anything that was deleted, we can insert instead of that wrong part, the right part, and vice versa. So we can have them reverted, right? Does it always happen? No, but we have the mechanisms to repair mistakes. Mistakes meaning mutations. But it seems that in our human body, only if the mutation has affected was an inserted base mutation, we can revert it. If it's deleterious, we can't. Right? Theoretically, we could. But in fact, we do not find deleterious mutations being reverted. Okay? So it seems that there's a difference between the two, where we do not take the liberty, meaning our cells do not take the liberty to dare to decide what was deleted, or maybe we have no mechanism of knowing what was deleted, but when something is inserted, we do. Okay? When a certain mutation causes a gene to lose its activity, to become inactive, we call that a forward mutation. Okay? So we had a gene, it was functioning, it was active, a forward mutation occurred in different mechanisms, right? We talked about different mechanisms of mutations. The fact that it made the gene inactive makes that mutation a forward mutation. Okay? And in a certain logic, a forward mutation can be reversed by a backward mutation. So we have an inactive gene due to a forward mutation that made it inactive, right? A backward mutation occurs and the gene becomes active. Okay? So back mutation have two basic types or subtypes. What we call true reversions, and the second one is what we call a second site reversion. Okay? A true reversion replaces the muted pair with the exact pair as should be. Okay, so let's say we have GC instead of AT. In this case, it will be repaired by taking out the GC, putting in the AT, right? A second site reversion does not restore the exact pair, but still makes the gene active and of worth. Okay, so we're not putting back the original pair that was there, but whatever was put in still works, still makes the gene active and functional. Okay. When we're talking about back mutations, they are thus more specific than forward mutations. Okay. When we kind of compare, what do we have more, forward mutations or backward or back mutations? The first kind is more than the other. So there are less back mutations than there are forward mutations. Let's say that we have a mutation in a gene. Let's say we have another gene that is able to cancel the effect of a certain mutation in another gene or within itself. The process of canceling that mutation, the effect of the mutation, is what we call suppression. So suppression is taking the effect of a mutation in a gene and canceling it. Okay? If we have a gene that is able to conduct that suppression process, we call it a suppressor. Okay? Now, if you write in Google or YouTube suppressor genes, you're gonna find that a more common term used is tumor suppressors, because that is the most likely process that we're going to want to suppress. So it will be probably related most likely to cancer, for instance. Okay, so in cancer we have the activity or inactivity, lack of activity of suppressors, and if a cancer cell is not suppressed, you're going to have cancer. All right. So you need those kind of suppressor genes to produce the suppression of tumors. Does that make sense? I hope so. Excuse me. So you are referred to a movie. Enjoy that. Okay. So is there a link between the size of the organism, of the species, and the amount of mutations that are going to occur, which will cause genes to become inactive? So the answer is yes. The bigger the organism, the more such mutations that make genes inactive occur. In other words, we find more mutations that make genes inactive in bigger organisms than in smaller. Okay? Or other larger species, let's say. It also means that genes may be targeted, lose their function, and be abolished. Okay? So let's take a whole gene, the whole sequence. And the next question is, are there certain sites on a gene, are there certain sequences that are more susceptible to mutations? Are they weaker? Do they have a bigger chance, a greater chance of a mutation occurring there? And the answer is yes. Okay? It doesn't mean that a mutation won't happen in another site. It just means that you can compare two sites on a gene, two sequences, and one may have a greater chance of having a mutation occur in it than the other. So basically, when we're comparing two sites, two locuses, right, we have three options. One, A has a greater chance for a mutation than B. The second option is that they have exactly the same chance. And the third option is that B has more chances than A. Okay? So, going back to the base pairing, when you think about it, since a pair has two bases in it, mathematically it gives you three options of mutations occurring at that same base pair. Okay? So let's say that we check out a gene called MSCEP. It doesn't really matter what it does. And this gene called MSCEP, which I have chosen just, randomly out of the top of my head, in most of the population has usually 100 mutations. And let's say that we compare MSCEP to other genes. Okay? Now we can see, does it have more mutations than the other, than average, less mutations than average, or the same? If it has more than the others, in both cases, those we will say that that gene has what we call hotspots. Hotspots are spots, locations, sites on the gene that are most likely to be affected by mutations. Okay? This hotspot may be affected or caused due to random or non-random mutations. Okay? Meaning that there are hotspots that have been conserved through time, through evolution, and those that are just occurring spontaneously. But they occur in that spot more than in others in average. Okay? Okay, we'll stop here and continue in the next video.